Hello, everybody. Welcome back. We are going to start jumping into our cellular reproduction chapters. We're going to be talking about cell division in both chapters 9 and 10. Chapter 9 is focusing on a cell division process called mitosis, where chapter 10 will be focusing on a cell division process called meiosis. So as we go through these processes, we'll be seeing some similarities and point out the differences between these two processes. So right here, what I have on this introductory slide is just a electron micrograph of a cell kind of towards the end of its cell division process. It probably started off as one cell, and then it divided its DNA, and it started to split, and then each of those two daughter cells would be identical copies of the parent cell. That's kind of the main point for mitosis. So let's go ahead and get started. As we get into this chapter, we need to talk a little bit about what cells do. So they don't spend their entire cell existence dividing. There's some time in between where they actually do cell stuff. So this is growth, this is expression of genes, maybe it's a secretory cell, you're going to be secreting maybe hormones or mucus, or maybe you're a muscle cell and you contract, or maybe you're a fat cell or a liver cell. You're just doing the things that you need to do. But there are some times where a cell needs to make more of itself for growth, for repair, for maintenance. We'll kind of list those things in a little bit. So then that's another part of the life cycle, if you will, of a cell. So a cell's just kind of growing and doing its stuff, but then it may spend some time dividing into daughter cells. Now, the main goal when you're dividing a cell is all of the genetic information, which is the blueprint to that for that cell to be what it needs to do, that has to be copied and distributed evenly and accurately to those daughter cells. So if you remember from our chapter 3, our biological molecules, the molecule of heredity is DNA. So that DNA that sits in the nucleus, the chromosomes, right, we'll talk more about those, they have to be copied and then split apart to go into those daughter cells. All right, so just a quick review of that structure of DNA. So we have the double helix. On the left here, we have shown just one half of it. So if you remember, we have a sugar phosphate backbone. We have A's, T's, C's, and G's are those nucleotide bases, those base pairs, A's with T's, C's with G's. And then over on the right-hand side, to kind of give you context, remember we have a picture of a cell, we have the nucleus, the DNA is in the nucleus, we have a chromosome. The chromosome is just a one big long piece of DNA. And what we have here in this picture, this is what I would like to call a duplicated chromosome. Because sometimes you see them as a single line, sometimes you see them kind of in this X shape. So whenever you see them kind of in this X shape, that is a clue that you're looking at a duplicated or replicated chromosome. It's two identical pieces of DNA that are stuck together in the middle here. So they are held together at the centromere. We'll be learning that word a little bit later. Okay, so why do cells divide? So there's lots of different reasons why cells might need to go through this process of mitosis. So I have a list here and I'm going to give you some picture examples. So we have organism growth. So here you plant a seed, the seed sprouts and it grows into a plant, all right? So organisms use mitosis to make more of these cells to grow in size. Mitosis is used for development, and a lot of time growth and development go really closely together. So here we see kind of the progression from a fertilized egg, a single-celled zygote. As it's continuing through this human fetal development, we have mitosis and mitosis and mitosis. You go from a single fertilized egg all the way up to a full functioning human with trillions of cells. And so that's definitely the organism growth part that we saw with the plants. That also happens similarly to humans. But we have development. We go from a ball of cells to a fully functioning, multifunctional organism with organs and tissues that all are doing very special things. We do cell division for for reproduction. So there's a couple different types of reproduction. We'll go into those a little bit deeper in a couple more slides, but this is just a very basic pictorial representation. You have cells going through asexual reproduction, which is just making an identical copy of themselves. So you can see in this orange cell here, its daughter cell is identical to the parent cell. So the parent cell started off with 4 chromosomes. The daughter cell has 4 chromosomes. Now, in sexual reproduction, the orange and the blue cells are representing the parents. And so this division right here, this is what we're going to call meiosis. So that's going to be chapter 10 stuff. So if we take a look at this parent cell, it has 4 chromosomes, and the daughter cell only has 2 chromosomes. And then same over here with the other parent. So it starts off with 4, we get 2. So then what do we do with our reproductive cells, the egg and sperm? We fuse them back together, and now we have an offspring that has 4 chromosomes. So we have different types of cell division depending on which type of reproduction is taking place. Mitosis will be used in asexual reproduction of eukaryotic cells. Meiosis will be used in the sexual reproduction of eukaryotic cells. Okay. Repair. So you scratch yourself. Oops, let me get rid of those arrows. They're kind of stuck on there. So when you damage your tissues, in this case, this looks like somebody scraped their elbow. So you have to repair that tissue. You don't permanently have that scrape. You might have a scar, but you will regrow your connective tissue. You will regrow your epithelial tissue. You'll cover it up, and sometimes you won't even know that that injury ever happened. So mitosis is a way, the cell division is a way to repair damaged tissues externally as well as internally. All right, and then lastly, we have day-to-day maintenance to maintain homeostasis. So I showed a picture of some of the organs of the digestive tract, your esophagus, your stomach, your small intestine, your large intestine. There's a lot of pretty abrasive stuff going on along your digestive tract. In your oral cavity, you're chewing, you're mashing food that could be pokey or sharp or hot or acidic. And then it moves through your digestive system where acids are attacking and enzymes are attacking, and this solid material is moving through. So those cells that line that surface of your digestive tract get beat up pretty often. And so we are constantly replacing those. And so this is what I like to call cell maintenance. So cells divide. They go through these processes of cell division, mitosis or meiosis, for a lot of different reasons. So the organisms grow and develop, they go through reproductive processes, they repair damage, and they maintain tissues. So that's why, that's kind of the end result of the processes that we're going to be going through in chapters 9 and 10. Okay, so the book talks a little bit about stem cells and this process of differentiation. So I thought I'd just spend some time on that, just a little bit, a little side note here. So stem cells are cells that have the ability to, when they go through cell division, they can become a different type of cell, kind of. There's some rules to go along with here. So here at the top of the picture, we have a stem cell, and it divides, and it kind of produces the bluish daughter cell on one side and the greenish daughter cell on the other. So when you are that zygote, that fertilized egg, and you go through multiple rounds of mitosis, you have a big ball of cells. All of those cells could be any cell in your body. They could be muscle or bone or blood cell or nerve cell or liver cell or skin cell. They haven't really been told what they're going to be yet. In the process of development, some of those stem cells actually get turned or differentiated into what they're going to be for the rest of its cell's life, for example. So that's that green cell pathway over here, this non-stem cell pathway. Get my pen back here, right here. So it's kind of like a stem cell in that when it divides, it will produce daughter cells, but of a particular type. So let's say you have a stem cell that once it differentiates, it will only be able to make more blood cells. And so once that differentiates, it can't go back and be able to make skin cells or bone cells. It's kind of programmed now to just be a stem cell specifically for blood. We have those all over our body. However, there's a few places in our body, not too much as we're an adult, a lot more when we are in fetal development and childhood development, but we have what's called self-renewal cells. So these are the ones that they can kind of maintain that blank slate, if you will. And so whenever they divide, they still maintain the ability to become any type of cell. That's really what researchers are talking about when we hear about stem cell research. They are trying to figure out ways to get these cells, these pluripotent or these potent cells, they have the ability to become any type of cell, because then they get to dictate what cell it's going to be. So if you get a petri dish full of these self-renewal stem cells and you need to repair a bone, then you can just give them the right signal and then they become bone cells, or they can be blood cells, or they can be nerve cells if we can do brain and spinal cord repair. So that's the big push in stem cell research, is trying to kind of not trick, but like give us the choice as to what those cells are going to be. There's some of these stem cells that are differentiated stem cells that can still be useful for preventing disease or fixing genetic problems and those types of things. So whenever you hear stem cell, that's kind of what that means. They're like blank slates. They have the ability to become a different type of cell, or if it's been differentiated, it might just be a differentiated stem cell, that it can only be one type. And then the product, so like a nerve cell, has lost the ability to divide. So it's no longer a stem cell, it's the final product. It's a nerve cell or a muscle cell, and it's lost the ability to divide. Some cells can maintain that, so bone cells can divide, skin cells can divide, but muscle and nervous tissue usually cannot divide after it's kind of reached its final identity, if you will. Okay, so just a quick mention about the reproduction processes for cell division. So chapter 10 is dealing with meiosis, which is going to focus primarily on the role of sexual reproduction. So I'm just setting the stage here for that. And this is, I guess, the human life cycle, if you want to call it that. So we'll start here with the fertilization event here. So you have your egg and sperm are fertilized. They produce this zygote that's going to grow up via mitosis into your adult organisms. Now, within what are called the gonads, those are the sex organs, the testes in males, the ovaries in females, their cells that have the total complement of 46 chromosomes, they go through that other type of cell division called meiosis. And in that process, they produce the sex cells. These are called gametes. Males produce sperm, females produce eggs. They have half the number of chromosomes, and that starts us all over again. So those gametes, the egg and sperm, will meet. You'll get fertilization. You'll get a diploid or a zygote that has 2 sets of each chromosome, grows up into the adult, and then the process starts over again. So we'll focus more on sexual reproduction in chapter 10. Compare that to some examples of asexual reproduction. This is where organisms use the process of mitosis, which is the focus of chapter 9, to make an identical copy of themselves. So here's bacteria is splitting. We have a paramecium is splitting. We have a hydra, which is a little, very simple animal that buds. So it just kind of makes a copy, almost like a photocopy of itself with the correct genetic information. And then these aspen trees, we have different groups of these aspen trees. They're kind of an interesting tree in that they have big extensions underground. So it's basically one organism, but then they can grow up the trees at different locations, and it's basically clones. So that would be an example of asexual reproduction, where the parents and the offspring are identical copies of each other. Okay, so let's take a look at some of our simpler cells, those prokaryotic cells. If you remember, they lack a nucleus, they have DNA, but they do not have a nucleus. So, and they're less complex. They don't have all the organelles that you see with a eukaryotic cell. They don't have all the linear chromosomes. They usually just have one circular chromosome. So in a bacteria or a prokaryotic cell's life cycle, it's mostly just growth and making a copy of your DNA. So the bacteria grow, they make a copy of the DNA, then they divide. And then those grow, they make a copy of the DNA, and then they divide. So it's a very basic cell cycle for prokaryotic cells. And they can do this very quickly. Some of the fastest cell division times for things like E. coli, which is found in your gut, or S. aureus, which is found on your skin and in your respiratory passageways, they can do their full cell division in like 20 minutes, which is actually pretty darn fast. Most bacteria take about 3 hours, where these guys are, replicating very quickly in about 20 minutes. Eukaryotic cells take on the hours to days time frame. So if you're trying to get some comparison, that's kind of how fast eukaryotic cells go through their cell cycle, their division processes, about a day or so. All right, so let's take a look at the process of what's called binary fission. This is that division part of that prokaryotic cell cycle. So you start off with step 1 is there's your bacteria. It doesn't have a nucleus, but it has its chromosome, one circular chromosome, and then it divides it, it copies it. So it replicates the DNA. So the DNA replication has happened. So now we have 2 identical copies of the chromosome. and they're both kind of attached to the inner surface of the cell. So then as the cell grows, those two copies of the DNA become further apart from each other. And then the cell, kind of follow the arrow up here to part 4, then the cell starts to split. We get a new cell wall, the plasma membrane meets in the middle, and voila, we are done. So they're calling this prokaryotic fission. You might also see it, come across it as called binary fission. We have 2 daughter cells that are exactly the same as our parent cell. They just might be a little bit smaller, but they have all of the chromosomal complement, all of the genes that they need to do their life stuff, their bacteria life. Okay. So that's prokaryotic cells. We're not going to spend too much more time on those. Eukaryotic cells, though, take a little bit more complex route to go from parent cell to daughter cell. So first, before we jump into that, we need to take a look at our DNA organization inside eukaryotic cells. So you might be familiar with the term chromosome. You've heard that before. Chromosomes are just basically the word for the big, long piece of DNA. The DNA is the molecule. The chromosome is the DNA, the whole piece of DNA. And a lot of times it is associated with proteins. They're called histones. These are the little yellow circles here. So the DNA double helix wraps itself around the histones. And so then you get like a little package of DNA. And then those packaged DNAs start winding around themselves into these coils. And then the coils start folding on themselves to make even more coils. So here we kind of have this loops. And they scaffold on these proteins. They kind of fold onto these proteins. And so then we have folds upon folds upon folds, and this big, long, stringy DNA molecule gets super coiled into this more condensed form when the cell is getting ready to divide. So that's really important because when you have your replicated DNA, that's, again, this is a duplicated chromosome because it looks like an X, right? So here's our X shape, held together in the middle here. That is a single chromosome that's already copied itself, held together at that centromere. If you were to stay long and loopy, like in this stage 2 or stage 1 or stage 2, when those chromosomes go to be broken and pulled apart, you could potentially damage that DNA because it's all coiled up and maybe bundled up with some of the other chromosomes. So in one of the first stages or one of the first steps of mitosis is this supercoiling, or what's called condensing of the DNA. What that does is it makes it really short. So when you do separate those replicated or duplicated chromosomes, you're not going to break anything off and lose some important genetic information. So when we talk about chromosomes condensing, imagine this picture. This is what they're doing. They're kind of supercoiling and folding and folding and folding to get to a nice dense collection of the DNA. Now, some terms here, we have the centromere. Like I mentioned, this is just this spot right in the middle. It holds the 2 replicated chromosomes together. And then at the end, they're showing those yellow bits. Those are called telomeres. So telomeres are sequences of DNA that help to protect the genes when a molecule of DNA goes to replicate itself. Okay, so here is our eukaryotic cell cycle. So remember that prokaryotic one was not very complicated. This one is a little bit more complex. So we'll take it piece by piece as we go through these different stages of the cell cycle, and then specifically spend a lot more time on the events of mitosis, the actual nuclear division. So some cells, they will divide regularly, like your skin cells divide regularly to maintain your skin and your lining. Some cells, once they get to a certain point, they're done. Like your neurons stop being able to divide, your skeletal muscle, your heart muscle, some of your cartilage cells stop being able to divide, and then that's it. So if you lose them, you don't get any more back. You might get some scar tissue, which isn't functional. It's not functionally the same. Some cells continue to go through this process on a regular basis, and some will divide when they're told to by growth factors or growth hormone telling it to, hey, it's time to grow or replicate or repair or something if there's damaged tissue. All right, so just an overview of the cell cycle. We're going to see this picture on some more slides. So the yellow part of the pie chart, if you will, so the yellow part of this cycle is an overall term that we call interphase. Right, interphase. And then the blue part is called, usually called the M phase, or that we're calling it the mitotic cell division part, phase. And so that's kind of what a cell alternates through. It's either an interphase, kind of doing cell stuff, or it's in the M phase. It's doing the actual nuclear division. So let's take a closer look at that interphase. So what's going on in this yellow band? So the interphase, and it's called interphase because early biologists saw it was what the cells were doing in between the times that they were dividing. So interphase is the phase in between mitotic phases. Even though interphase lasts the longest, mitosis is actually a very short time piece of the entire cell division, cell cycle. So the interphase part of the cell cycle is divided into three smaller phases, if you will. They're called G1, S, and G2. So the G, you can call it growth phase. Some other sources might call it the gap or the first gap phase. So G1 is a growth phase. And so this is the cell is acquiring nutrients. It's growing to the proper size before it has the ability to replicate or divide itself. And it is doing the cell thing. So it's just being a liver cell, being a kidney cell, being a skin cell, whatever it's doing, if it has that ability to do what it's doing. So then somewhere in the G1 phase, there will be a signal that say, hey, you got to get ready to divide. We're going to split into two daughter cells. So then that's going to be the transition into the S phase. S stands for synthesis. DNA replication takes place here. So the S phase of interphase is the DNA is replicated. Every chromosome is replicated, and it goes from that single strand of DNA to that X shape, the replicated strand with the centromere in the middle. So the S phase is the beginning of the preparation, if you will, for mitosis. We're replicating the DNA, so all of the daughter cells will, or both, I should say, both of the daughter cells will have identical copies of that genetic information. All right, so then the DNA replication gets done, and then we move out of S phase into G2. So G2 is our second growth phase, or gap 2. And so this is the final preparation for getting into the mitotic phases. So we are making more organelles. Maybe we're replicating our mitochondria or chloroplasts for a plant cell. We're making more endoplasmic reticulum. We're making more vesicles. We're making more lysosomes. All of those goodies that you learned back in chapter 4 of cell structure, we have to make enough of those so when the cell does split, the daughter cells will have a full complement of everything they need, because mitosis will be getting ready for the dividing of the nucleus, where G2 is getting everything ready. So there's enough stuff to go around. Okay, and then so this last time we're going to be seeing the cell cycle is taking a look at the blue part, right? So the yellow was interphase, G1, S phase, G2, that's all under the umbrella of interphase. And then the blue part is kind of broken up into mitosis, which is prophase, metaphase, anaphase, telophase, and then cytokinesis. So I know I don't really like that they have the and here. I would put telophase in its right over here, right after anaphase, because cytokinesis is technically not part of mitosis. So mitosis is the processes that take place to actually divide the nucleus. It's nuclear division. We're pulling apart those replicated chromosomes. Cytokinesis, right? So in G2, we prepped everything else. We have enough organelles and things. Cytokinesis is actually the physical splitting of the cytoplasm and organelles, which comes after all of the stages of mitosis. Okay, so let's take a quick look at the eukaryotic chromosome. So again, a chromosome is a long piece of DNA. We saw it in that earlier slide where it kind of supercoils. So this is a very basic schematic of it. It's not showing the double helix form, but at least you kind of understand that's DNA in there. So the blue mark in the middle is representing the centromere. So that's a non-coding place. It's kind of a dense area. It's got some proteins associated with it. And then the colored bands are the genes. So we have a kind of a gray, a purple, a dark green, and a light green. So those genes are representing instructions for particular cell products. It might be the gene that codes for hemoglobin, or the gene that codes for insulin, or the gene that codes for how curly your hair is, or what color your eyes are, or whether your earlobes are attached to your head or if they're floppy. So that's what genes are. That's the heredity stuff that we'll be getting into. So the eukaryotic chromosome is a linear one piece of DNA. In the process of S phase, it is going to copy itself. It does the DNA replication. but it stays connected in the middle at that centromere. So when we are taking a look right here, that is the connection point of the two replicated pieces of DNA. They are identical. So the gray gene is identical to the gray gene on the other side. When we have it in this form, again, in that X form, right, this is what we call a duplicated chromosome, or replicated. You might hear it that way. I kind of go bounce back and forth between both of those words. So we have two identical copies of DNA held tightly together at the centromere. And just to kind of make things more complicated for biology students, right, let's call these bits of DNA something new. So these are what we call sister chromatids, right? So an individual chromosome up here, just one individual chromosome. When that individual chromosome replicates, it's still one chromosome. It's got all of the same genes on it. We're not making a completely different chromosome. We are just copying it into its partner, which is a sister chromatid held tightly together at the centromere. And then at the end of mitosis, those replicated chromosomes that's held together at the centromere will be pulled apart. And that's what we're seeing here is now we're back into our independent individual chromosomes that are identical to each other. So we start off with one, we replicate to two, and we split back up into two separate chromosomes at the end of mitosis. So keep that in mind when we're looking at these pictures of mitosis, and again in meiosis as well. Okay, so we're going to jump right in and take a look at the phases of mitosis. We're actually going to start with interphase, which again is not technically part of mitosis. It's all of that yellow stuff. But when we're looking at a cell, we can't really differentiate between the G1, the S, or the G2 phase of interphase. We could just see it as an interphase cell. So here we have the typical looking cell. You've got the plasma membrane, you got the readily identifiable nucleus, you got a nice dark nuclear envelope. We've got all the DNA is still kind of loosely coiled. It hasn't started super condensing yet. And we have that structure of the nucleolus. If you remember from chapter 4, that's the location of ribosome assembly. Okay, and then we have some centrioles. So centrioles are those organelles involved in DNA or cell division. And here we are. So this is the pictorial representation of interphase. This is an animal cell representation. So we can see our plasma membrane. Let me get a brighter color here. We can see our plasma membrane. We can see our nuclear envelope. We can see the nucleolus. And all that purple stuff is just that loosely coiled DNA. Sometimes it's called chromatin. You might hear that word come around if you're watching videos or animations. So chromatin is just a loosely coiled DNA. Oh yeah, there it is right here, chromatin. It is not folded and folded like we're going to see in prophase. So a cell here, we would not be able to tell whether this cell is in G1 S phase or G2 phase. We'll just call it an interphase cell. Okay. All right, so let's jump in. So let's say the DNA has been replicated, G2, we've got enough goodies for everybody to go around when it splits. So we are physically now starting mitosis, which is the first stage of mitosis is prophase. So lots of resources divide this up into sometimes called early and late prophase or prophase and prometaphase. I just bundle it all together in prophase. So just listing all the things that happen in early and late. So our replicated chromosomes that replicated in interphase, S phase of interphase, they start to condense. So here we can see that condensing chromosomes. So the loosely organized DNA is now starting to do all that supercoiling. So we can actually start visualizing the chromosomes in the nucleus as individual blobs of dark stuff. Right? So we still can't really single them out quite yet. The spindle starts to form, so those centrioles are starting to move to opposite ends of the cell. These microtubules start to grow from centriole to centriole, kind of going towards the middle of the cell. The nucleolus breaks apart, just kind of disassembles, because we're not actively putting ribosomes together during mitosis. The nuclear envelope breaks down, so it's starting to break down in early phase, prophase, and it is completely gone in late prophase. And then the microtubules will start attaching to the centromeres. So that kind of connector point where the two sister chromatids are connected, these spindle tubules come in and connect to theirs with special proteins called kinetochore proteins. I think we'll see that in the next picture, or we see it right here. Kinetochores are kind of proteins that are found at the centromere that allows these spindle fibers to connect to the centromere, the middle part, kind of that non-coding piece of DNA. And eventually we're going to see that that actually gets pulled apart. So there's a lot of things happening in prophase. So the chromosomes are condensing, the nucleolus disappears, the nuclear envelope disappears, the spindle is starting to form, and the microtubules are starting to attach. This as the spindle is growing towards those chromosomes. All right, so that's prophase, the first part of mitosis. All right, so then in metaphase, the chromosomes are completely condensed, the nuclear envelope is completely gone, the spindle is all formed by the end of prophase. All of our replicated chromosomes have been attached by these spindle fibers, these microtubules attaching to the kinetochore, and they're kind of almost like a tug of war. They're pulling back and forth, and eventually what they'll do is they'll line all of the replicated chromosomes up on an imaginary middle line called the metaphase plate. And so here we can see that kind of across this cell right here. So just kind of drawing that dotted line, that would be the metaphase plate. The spindle fibers are kind of pulling back and forth until we get all of those replicated chromosomes lined up, and that's metaphase. I always remember that as metaphase lines up in the middle. They both start with the letter M, a simple way to remember, but that might help. Okay, so metaphase, we move on to anaphase. So anaphase is all of those replicated chromosomes that had the spindle fibers attached to their kinetochores. Those spindle fibers start getting shorter. And what they'll do is they'll pull and they'll tug, and eventually they will break apart those replicated chromosomes into the individual chromosomes, and those spindle fibers get shorter, and it starts to pull those the sister chromatids have been pulled apart. Now they're considered individual chromosomes, and they get pulled to opposite ends of the cell. So this is our anaphase, and I remember anaphase because the chromosomes are pulled apart. A for anaphase, A for apart. Another simple way to remember that one. All right, so we're being pulled apart, and we can see this again here in this animal cell. So here's our plasma membrane. It's going to outline the cell itself. Here's where our centrioles would be, kind of at the ends of all these spindle fibers. All of kind of the pink lines are the spindle fibers, and the dark purple lines are representing those separated chromosomes being pulled to the opposite end of the cell. All right, the last phase of mitosis is called telophase. So telophase is almost like the reverse of prophase. I like to think of it that way, is as we were building the microtubules for the spindle in prophase, we are breaking those down. Where we lost the nuclear envelope in prophase, the nuclear envelope starts to reform around those newly separated chromosomes. Where the chromosomes were condensing in prophase, they're starting to unwind in telophase and go back to that chromatin form. Where the nucleoli disappeared in prophase, the nucleoli come back in telophase. So just if you know all the events of prophase, just reverse those for telophase. So that kind of helps with the studying. So again, here is our outline of our animal cell. It's still considered one cell. It has not completely split. This is still telophase. This is where our new nuclear envelope is going to start forming around our newly divided chromosomes. So that is our telophase. And that finishes mitosis. So we have successfully divided the nucleus, but we haven't divided the whole cell yet. So that takes us on to cytokinesis. So in animal cells, which we've been using as an example, cytokinesis actually starts pinching because it's flexible, right? They don't have a cell wall. So it kind of pinches and produces what's called a cleavage furrow, which is kind of this area right here. Okay, so cleavage furrows on both sides. Some little contractile proteins go in and just kind of physically cinch and pinch the middle part together, and it separates the cytoplasm and the organelles. So at the end of cytokinesis, you'll have two completely separate identical daughter cells. All right, so there's an electron micrograph of that stage of cytokinesis. Okay, so cytokinesis in plants is different than in animal cells because plant cells have cell walls, right? So they can't do the pinching like you saw the cleavage furrow. That doesn't exist in plant cells. So instead of, since they can't pinch their rigid cell wall, they actually use the Golgi, start sending vesicles filled with cell wall material to that central area. And so what that's going to do is to physically separate the two new daughter cells, even though they're kind of contained within that original cell wall. So that's how plant cells complete the process of cell division with cytokinesis, is they create a new cell wall. As it's growing, it's called the cell plate. And then once it's completely formed, it's just cell wall. Okay, so the processes of mitosis, they're great for all of those functions that we need to do, right? So we have to grow and develop and reproduce and repair and maintain. But there can be some problems that could arise from too much mitosis. So we have to regulate that. And so there's some special regulatory mechanisms your book mentions just briefly. We'll call this the control of the cell cycle. So some cells, like I mentioned earlier, like the lining of your digestive tract, your skin, your bones, they have the ability to maintain this reproductive properties. They can actually go through mitosis, and they have to be regulated. Where other tissues like the brain, the heart muscle, the skeletal muscles, some of your cartilages, they've lost that ability to replicate. And so they are kind of turned off. They're in what's called a dormant state. But the cells that can replicate, if they replicate out of control, if there's no stop button to say, hey, stop reproducing, this is what leads to cancer. So I know your book doesn't talk too much about this, so this is kind of my kind of life application, is basically what cancer is. The tumors that you have or the blood cancers that you have, they're just uncontrolled mitosis. There was a mistake somewhere in these control checkpoints that causes the tissue to grow out of control, and that creates the solid mass of cells, which is what we call cancer. So we have these checkpoints, and I have a diagram. It's not in this new edition, but you can find it here on the PowerPoint, or you can find on an internet search. But very basic level we're going to introduce here. There are checkpoints that the cell kind of does like a look at yourself, saying, is everything okay to move on to the next stage? So there's three major checkpoints we're going to take a look at: moving from the G1 to the S phase, moving from G2 into mitosis, and the going from metaphase to anaphase. Okay, so here's a diagram that kind of goes with those three checkpoints. They're kind of illustrating it like a crossing guard, saying, whoa, you can't go, or it's okay to go. So let's say we are moving kind of in this direction. So our cell is in G1, and if you remember, G1 was growing, it was acquiring nutrients, it's making sure it's big enough. And so when it gets the signal that it's going to need to divide and replicate its DNA, there's some chemical communication messengers going around within the cell that says, hey, before I'm going to let you go into S phase, is your DNA intact? Because if we're going to replicate, we want to make sure that everything is normal. There's no problems. We're not missing any pieces of DNA there. It's not a significant amount of mutations, and it's okay to make a copy of that genetic information. If in the process of the self-check, things come back as saying, no, we're missing some of our DNA, then it won't be allowed to go on to the next phase, because we don't want to replicate a cell that's got problems. We don't want to replicate the DNA if there are problems, because then that will get perpetuated into the next generation of cells. So this G1 checkpoint says, no, you cannot go on until your DNA is fixed. And sometimes they can go back and fix it, or the cell just might say, get a signal saying, okay, you're kind of messed up, you're going to die. You're going to go through what's called apoptosis, which is programmed cell death, because there's too much errors in the DNA, and we do not want that to be sent on to the next generation. All right, so if everything's good, we will progress into our S phase. We will replicate our DNA, and then we will go into the G2 phase. And before we can jump into mitosis, this G2M checkpoint says, all right, before we go into mitosis and actually divide your DNA, is the DNA replication complete? And is it accurate? We're not missing anything. So again, it's another checkpoint for that genetic information. Before we go and expend all this energy, we don't want to go into mitosis if we have mistakes with our DNA. All right, and then the last checkpoint is the metaphase, anaphase. So let's say everything's good. With our replicated DNA, we go in here, we do all the prophase stuff, we line everybody up on the metaphase plate. And so this checkpoint says, Are all of the replicated chromosomes lined up on the metaphase plate? Do they all have connections to the microtubules at the kinetochore? We don't want one whole replicated chromosome going to one side because then the daughter cell will be missing its copy. So that checkpoint makes sure that all of your replicated chromosomes are lined up and that they all have attachments to the kinetochore. And if everything is okay, we get the green light at every time, then the cell is going to continue to go through that process, and then it will be successful in producing two identical daughter cells. If there are problems, so if the checkpoints come back, these kind of circulating proteins saying, no, there's not a problem, or sorry, no, there is a problem, then it will not progress through the checkpoint. Now, with cancer, a lot of times what happens is the mutations that cause cancer are these checkpoint proteins. So the proteins that say, stop before you're to see if your DNA is intact, stop before to see if your DNA is replicated, that's the problem. And so there could be mutations in your DNA or problems with your cell, but the checkpoint proteins aren't working and the cell continues to go through replication normally when it would not have. And so that's kind of the key basis for cancerous cells, is they've lost their ability to regulate their cell cycle. So that's kind of an important connection to what a lot of you may have personal connections with in your lives or people that you know. Okay, so that wraps up our lecture on mitosis. So again, go through the stages, make sure you're comfortable with the order, with all of the parts and pieces, the names like the chromosomes, the kinetochore, the centromere, the sister chromatids, what happens in each of the phases, prophase, metaphase, anaphase, telophase. And then we're going to end here with kind of like a little pop quiz of, I'm going to show you some pictures of the animal cells, and then I'll show you pictures of some plant cells, and see if you can identify some of these phases, like if you were actually in class looking at these under a microscope. So I'll give you a few minutes to look at this, or you can pause the video, or you can just go back and take a look at the PowerPoint and see if you can find as many of the different phases of the cell cycle as you can. So I'll get my pen ready. Let's see, let's do purple. It's kind of a cool color. All right, so some of these might be pretty obvious, so maybe you've paused and you're coming back, but where my eye goes right away is this one right here. So you see that one? So what phase is that? Take a look at what's happening with the chromosomes, what's happening with the shape of the cell. Those are going to be your biggest clues. This is a representation of telophase. So here we have our cleavage furrow right here. We have our two separated bits of DNA that will then be surrounded by a nucleus as the telophase processes finish up. All right, so let's see, what else do we have here? Another really obvious one that my eye is drawn to is this one down here at the bottom. Kind of circling that with the purple pen. So which phase is that representing? Look at the chromosomes. Where are they headed? What are they doing? This is an example of anaphase. So anaphase, we can see the chromosomes kind of being pulled apart, the replicated chromosomes being pulled apart from each other. They kind of have like this, almost like this perspective here. So that's a good anaphase. Let's see, what else? Right next door here, let's take a look at this one. What phase does that look like? Again, where the chromosomes are, that's going to be your biggest clue. So we have some condensed chromosomes. They look like they're all lined up on the middle of the cell. There's your clue. That is a good example of metaphase. Okay, and I'm looking around. So let's take a look at this guy down here in the bottom. This one right here. There's a lot of these actually in this particular slide. So what phase, I'll say, of the cell cycle, so that's kind of a clue, what phase is that one? Well, if we take a look, we don't really see any obvious chromosomes. Nothing is lined up and arranged in a very special way. So that is kind of your biggest clue. If we can't see the visibly condensed chromosomes, this is a representation of interphase. The cell is in interphase. We cannot tell whether it's G1, S, or G2, but it has a clearly defined nucleus. Those darker specks in the middle are the nucleolus, and so those are all clues that we are looking at interphase. The one cell phase I couldn't find on here is prophase. This one might be prophase, but I'm not sure, so I'm just going to put a big question mark in that one. Not sure. That's okay. All right, so let's see if I can get to the plant cell. Oh, I got to get rid of all my markers here. So as I am erasing these, kind of take a look at the plant cells, see if you can identify some phases of mitosis in the plant cells. Maybe I'll get a green since purple might kind of fade in. All right, I think we have all phases in the plant cell. So we know it's plants because it's very bricky looking. We have clear defined cell walls. So I'm actually going to go in order. So hopefully you pause and try to figure it out yourself. So I'm going to go in order of the cell cycle. So a really good interphase would be this one right here, over here, kind of right above my head. So we can see a clearly defined nucleus, and we can see two nucleoli. So again, we can't see individual chromosomes. It's like chromatin form, a nicely formed nucleus, and visible nucleoli. So then after interphase was prophase. This guy up here towards the top, I'm kind of outlining in the green right now. This is a good example of prophase. We can start to see some coiled condensed chromosomes. The nuclear envelope is kind of breaking up a little bit, and the chromosomes aren't lined up and they're not being pulled apart. And we don't see a cell plate. So kind of by process of elimination, that's a good example of prophase. All right, and then after prophase is metaphase. So we're going to be looking for chromosomes kind of lined up in the middle of the cell. Now, plant cells are a little bit, their chromosomes are kind of long and pokey. So this is actually a good example of metaphase right here, kind of in the middle of the screen. So all the chromosomes are kind of lined up here, even though they might have some of their chromatids sticking off towards the poles of the cell. So that's our metaphase. And then what do we have? Anaphase comes next. So hopefully you see right next door to the metaphase, here is our anaphase. All right, so we can see those chromosomes that were lined up with the metaphase plate are now being pulled apart to opposite ends of the cell. And then lastly, see if you can find telophase. Telophase. So I see a good one right over here. This is our telophase right here. So telophase in plants, the good clue is kind of late telophase, early cytokinesis, because they kind of progress one right into the other, is that our chromosomes are starting to, they're on opposite ends of the cell. They're starting to uncoil, so they're not individual chromosomes anymore. So the nuclear envelope is starting to form right here. But another clue is we're starting to see the very beginning of that cell plate right here. A very subtle line right there. And then my guess, I'm going to pop over here, is that this was a cell that just finished cytokinesis, right? So we can see our nucleus, we can see the nucleolus starting to show up, and we have a pretty defined cell wall at that point. All right, so with this one slide, we got to see all of the phases of the cell cycle. So interphase, prophase, metaphase, anaphase, telophase, and a cytokinesis. So, all right, so practice your stages of mitosis, and I will see you next time for meiosis.